Okay, well, my watch is reading 4 o'clock, so we're going to get started, and I am absolutely stoked today for this talk. But we've got some slides to get through first before we can get to it, so let's get to it. So if you don't know much about Crow Canyon, we are a nonprofit located in Southwest Colorado, very close to Mesa Verde National Park. If you want to learn more about us or our mission, you can visit crowcanyon.org. That is our website, or you could email info@crowcanyon.org. We are all pretty well versed with Zoom at this point in time, I believe. But I just want to say, if you have a question, do not put it in the chat. Put it in the Q&A section that is circled in red here on this slide. If you miss anything, I think this talk will likely be up on YouTube, and so you can look within the next, 24 to 48 hours for that, I believe. Oh, messed up here. Be sure to do a land acknowledgement. So the Crow Canyon Archaeological Center acknowledges the Pueblo, Ute, Paiute, Dene, and Jicarilla Apache, people on whose traditional homelands our campus sits. With the work we are doing at Crow Canyon, we seek to bring light into the darkness of memory that these lands, waters, wildlife, and her descendants hold as a result of policies of removal, genocide, and displacement. We seek paths that support and align with Indigenous people and communities. With gratitude, we honor the contributions of present-day Indigenous communities, descendants, and colleagues with whom we partner, who have helped guide our work and our mission. If you are interested in maybe helping support the webinar series here, that is the loving product of our late friend Taylor, you can scan this QR code here and learn how to maybe donate to the webinar series. I'll leave it here for just a beat, just in case someone is really excited to do just that. And we're going to go on. Okay, we are really excited at Crow Canyon to announce that our cultural explorations team has our 2026 schedule open now. We've got 4 awesome programs here that you could go to our website and check out. Just some of them is Mesa Verde and Beyond Cycling, the Ute Mountain Ute Tribal Park. Another one, Monumental Decisions, the Antiquities Act of 1906. Some really awesome programs. We have 2 upcoming webinar events next week and then the following week, so be sure to check those out and register for those. And finally, we're going to get to introducing our speaker today. So Dr. Emma Elliott Smith is a historical ecologist who integrates zooarchaeological records, natural history collections, and contemporary field data to reconstruct food webs and assess how human activities and environmental change have shaped modern ecosystems. By measuring isotopic ratios preserved in archaeological and museum specimens, her research pushes ecological baselines centuries beyond written records, giving archaeologists and conservation biologists a quantitative framework for evaluating long-term resource use and ecological resilience. So without further ado, we're going to turn it over to Emma.
All right. Okay. All right. Is my screen showing?
Yes.
Excellent. All right. So I'm going to jump right in. Um, Thank you so much, John, for that introduction. And I also really want to thank Benjamin, Laura, and Taylor, and really everybody at Crow for the invitation to speak here today. And of course, I'm also incredibly excited at everyone who's with us online because I get the opportunity to share some of my ongoing research integrating zooarchaeology and conservation biology. And I put as many gratuitous pictures of cute sea otters as I could into this talk. So hopefully it will not disappoint in that regard. Okay, there we go. But first, I need to acknowledge just some of the many people who have contributed to this work. The kind of interdisciplinary research I'll be talking about today is really only possible through collaborations. And I feel incredibly privileged to have worked with a kind of unique blend of anthropologists and biologists, as well as some isotopic geochemists. And then I also just want to take a minute to give a special acknowledgement to Dr. Jim Estes, who sadly just passed away this May. Jim, if you don't know who he is, he had a really tremendous impact on how we think about food webs and species interactions. He's particularly famous for his work on sea otters, which is how I got to know him. He was actually my very first research mentor, who I met when I was like a sophomore, bright-eyed, enthusiastic undergraduate. And he was incredibly generous and invited me into his laboratory, and that's why I'm still here working on sea otters 15 years later. So I know he would have been really excited about the data in this talk. So thanks for everything, Jim, and we miss you and think about you a lot. Okay, so John, from my bio, there's a lot of words in there, a lot of jargon-heavy words in there. So I just wanted to take a minute and sort of explain to everybody what I do, because I know that this talk is probably a little bit different than some of the webinar series that Crocanian has seen before. So my formal training is not in anthropology, but in biology, and specifically in ecological sciences. So, I got into it from sort of that perspective, but I realized pretty quickly that ecology has a really big blind spot, and that is time. So most of what we know about food webs and species interactions come from studies that have taken place over the last 50 years. And of course, a lot had already happened by the year 1970, right? And so this means that we've been studying ecosystems that are already heavily impacted by predominantly Euro-American industrialized activities. And this realization is kind of what pushed me towards looking deeper into the past. Because if we want to understand what's possible, we need records that reach further back than our modern ecological monitoring does. And so that's where zooarchaeology, and in some cases paleoecology, really comes in. Because what these records are basically time machines, right? They preserve bones, shells, artifacts that record not just the life histories and ecologies of animals, but also the way that people were interacting with these animals and with ecosystems more broadly. And then to connect all of this, sort of the major focus of my research is using a type of chemical analysis known as stable isotope analysis, which I will talk your ear off more about a little bit later on. And what this does is basically just give us a common currency for comparing the past to the present. So whether the sample comes from a living sea otter, an archaeological bone, or a museum specimen collected 100 years ago, we can speak the same language, the language of the elements of carbon and nitrogen and hydrogen. And this allows us to bridge these different time periods and different spatial and temporal scales and really trace how humans and ecosystems have changed together through time. Okay, so on to the main subject of this talk, which is sea otters. So I know there aren't a lot of these guys in either Colorado or New Mexico, where I'm currently based. So I wanted to start with just a brief primer of the ecology of sea otters, their natural history, their lived history, and why you should care about them and their conservation. So as is hopefully obvious from the name, right, sea otters are marine mammals. But it might be better to actually say that they are coastal mammals. So this top figure here in the purple band shows the geographic distribution of sea otters as of 2015. We're going to come back to this graph again in just a few slides here. But what you'll notice, not a lot of sea otters out in the middle of the Pacific Ocean, right? They're really restricted to a narrow range just offshore. But throughout this distribution, they're found in a very wide array of habitats. And some examples are shown in the photos here on the bottom. Sea otters are very famous for their association with kelp forests. So the sea otter on the left is resting in a kelp forest bed off the coast of Washington, but they're also found in many soft sediment environments. So seagrasses, seagrass ecosystems, estuaries, just to name a few. And sea otters are also unique in that they are absolutely voracious carnivores. So I think of them as the hummingbirds of the sea because they have incredibly high mass-specific metabolic rates. And unlike other marine mammals like your seals, sea lions, or whales, sea otters don't have blubber. So they don't have this insulating layer to help keep them warm. They do have incredibly thick fur, but most of the way that they keep warm is by eating pretty much constantly. And what they eat is about any type of macro invertebrate, so, large invertebrate that they can get their paws on. So some examples here, we've got a sea otter eating a crab, a sea urchin, clam, abalone, an octopus, and down on the right there is a sea otter eating a sea cucumber. So at the population level, sea otters are thought of as macroinvertebrate generalists, but there can be a lot of interesting variation among individuals within a population. And because sea otters eat so much, they actually play an incredibly outsized role in shaping and stabilizing coastal ecosystems. So sea otters are what's known as a keystone species in ecology. And this means that even in really small numbers, they can completely transform and stabilize coastal habitats. So the classic examples are from work by the late Jim Estes, and it's kind of highlighted in this graphic here, where you see on the left, when sea otters are present, they're eating a lot of those sea urchins, those little purple spiky dudes, so that the sea urchin numbers stay low and kelp forests are able to grow and survive and thrive. When sea otters are absent, sea urchins overgraze the forests, and these whole ecosystems collapse into what's known as an urchin barren. And if you're not familiar with sea urchins, by the way, they're like little underwater cows. They graze constantly, and without predators like sea otters to keep them in check, they can mow an entire kelp forest down to bare rock in a very short amount of time. We've also learned more recently that this phenomena is not just present in kelp forests. So more recent studies have shown that sea otters have similar stabilizing effects on seagrass meadows and estuaries. So their influence extends well beyond the rocky coastlines where we usually picture them. So apologies if you hear my cat meowing in the background. Um, but okay, so this is sort of sea otters and their ecology and natural history. But their story isn't just ecological, it's also deeply human. And our relationship with sea otters is long and pretty complicated. So it started, of course, in North America with indigenous communities. So indigenous peoples of the Northeast Pacific coast have deeply rooted traditions of sea otter harvest and management. The archaeological record suggests that this stretches back at least 10,000 years, if not more. And these traditions of hunting and managing sea otters are part of a broader system of stewardship that tied indigenous peoples and all their coastal habitats together. And this is one reason the archaeological record of sea otters is so rich. So why were people hunting sea otters? Well, it probably wasn't for food because there really isn't a lot of meat on a sea otter. And as we've just talked about, they don't have blubber. What they do have are these incredibly dense pelts. So a lot of why people were hunting sea otters was for these pelts, which have been and continue to be prized for their warmth, beauty, and ceremonial use. So on the top right here is an example of some mittens that were made by indigenous communities in Alaska using sea otter pelts. But we also think that people were harvesting sea otters not just for their fur, but in some areas to keep them out of managed intertidal areas. So the photo on the left here is showing what's known as a clam garden in British Columbia. And these are managed intertidal areas that people were really important to people for their local food security. And so it's thought that sea otters were often hunted out of local areas to keep them from raiding clam gardens. And I also, of course, would be remiss if I didn't mention that these relationships haven't ended, right? So traditional harvest of sea otters continues today in parts of Alaska and British Columbia, forming a really important link between this cultural heritage and our modern conservation discussions. Okay, and what's another important point here is that, in spite of this millennia of harvests by indigenous communities, until the 1600s, sea otters were found in a nearly contiguous distribution all across the North Pacific Rim. So you can see in this yellow band here, we're pretty sure sea otters ranged from northern Japan all the way down into Baja California. Excuse me. And then, of course, the story changes dramatically when Europeans and Euro-Americans arrive onto the scene in the 16 and 1700s. And the sea otter's remarkable fur attracted them as well. It became a global commodity, and the maritime fur trade exploded almost overnight. And unfortunately, indigenous communities were often coerced and forced into this trade, hunting sea otters under really violent colonial systems. And at the height of this industry, it was absolutely devastating. So the graph I'm showing here shows the number of sea otters that were removed from just Alaskan waters over, about 150 years. And the tallest bar you note here was for 3 years, from 1804 to 1807, and there was more than 15,000 sea otters that were removed just from Alaska alone in those few years. And as a result, by the early 1900s, there were fewer than 2,000 sea otters left in the world across their entire historic range. So they were effectively erased off the map. So the red circles you can see here are where a few remnant sea otter populations managed to hang on during the fur trade. Fortunately, in the 20th century, efforts to protect and bring back the species got underway. So there was a series of landmark legislations in the early 1900s that effectively ended the maritime fur trade. And then the black arrows on here show sites where in the 1960s and 1980s, US and Canadian scientists began translocating sea otters. So they took them from these remnant populations and brought them to other parts of their former range. Some of these reintroductions, which we're going to talk about, like that in Southeast Alaska, did really well, whereas other ones, such as Oregon, really never took hold. And so today, this purple band is showing where we find sea otters in the present. Actually, about 10 years ago, but it's not too much different than it was back in 2015. So in large parts of their range, mostly in the Aleutian Islands, southern Alaska, and parts of British Columbia, sea otters are back into their historical distribution. But we have some big gaps, most notably from southern Washington all the way to Monterey, California. We still lack sea otter populations in the region. And this patchy recovery also raises another key question for me, which is what does recovery actually mean, right? So counting animals and mapping ranges is important, but it's only part of the story. And we also want to know whether sea otters have recovered their ecological role as they've recovered their geographic distribution. And that's really why we need a historical perspective. So by turning to the archaeological record, we can reconstruct what sea otters were doing before industrial exploitation. So we can ask which habitats they used, what they were eating, whether this varied geographically, and how they were interacting with their coastal ecosystems and also with local people. And once we have these really critical baseline data, then we can actually start to evaluate whether sea otters have recovered into their historical ecological niche. And if they haven't, why not? And can we fix it? All right, so this all sounds great, but now we come upon a really important question, which is how on earth can we know what an animal was eating and where it was living 100 or 1,000 years ago? And as I alluded to in the introduction of this seminar, this is made possible through chemical analysis of well-preserved archaeological specimens. So this is an example of some sea otter femora that I've sampled from an archaeological site in southeastern Alaska. And what's amazing is that in a lot of these bones, the original organic material from when that sea otter was alive is still present. And if it is present, then we can analyze the chemistry of this organic material and make inferences into the ecology of the animal. Because at a fundamental level, we all are what we eat, right? So we can take advantage of that. All right, so how does this work? Well, now we're getting into kind of the nitty-gritty of the method that I use in my science, which is known as stable isotope analysis. And I hope that you will forgive me if I drag you briefly back to Chemistry 101 so that we can all kind of get up to speed on how this works. So what you probably remember is that elements can have different versions, and those versions are known as isotopes. And isotopes are defined by having a different number of neutrons in the nucleus of an atom. So for example, almost all carbon here on Earth has a mass of 12, meaning it has 6 protons and 6 neutrons. But some carbon atoms, about 1%, again here on Earth, have an extra neutron, which makes them a little bit heavier, giving them a mass of 13, hence the name carbon-13. And I also want to note that this type of carbon is stable, unlike its more famous cousin, carbon-14, which of course is radiocarbon and is used for dating. And the really awesome thing about this from an ecological standpoint is that this is a really small mass difference. So, it doesn't change what bonds these elements form. Carbon is still carbon, but it does yield different thermodynamic properties. And so these isotopes move at different rates through biological and chemical reactions. So as plants and animals are living and building their tissues, they're going to incorporate different amounts of these stable isotopes depending on what they're doing, what they're eating, what habitat they're living in. And these signals can, in some cases, be preserved into the fossil and zooarchaeological record. All right, so what I do is I measure the ratio of heavy to light isotopes in a sample. And then we all have to calibrate this relative to an international standard so that I know my data from my laboratory are directly comparable with somebody, at Wyoming or in Europe. And we report everything in what's known as delta notation, which are these squiggly looking Ds here. And I'm going to be showing carbon and nitrogen isotope data today, which means we're looking at these ratios here. So 13 to 12 carbon, 15 to 14 nitrogen. And in exactly the same way that you could calculate a percentage from parts per hundred, we report isotopic data in per mil, which is simply just parts per thousand. And then the last point I want to make here is that carbon isotope values, because we're comparing everything to this international standard, carbon isotope values are often negative. So this is very confusing, understandably, for people, and I just wanted to make a note of why that is. Okay, and then we often look at isotopic data in this sort of bivariate space where carbon isotopes are on our x-axis and nitrogen isotopes are on our y-axis. And we do this because this biplot can kind of be thought of as an ecological map. And each of these axes represents a key dimension of organismal ecology. So for instance, carbon isotope values tend to differ among types of photosynthetic organisms, which are often called primary producers. So when you hear me say primary producers, I mean something like a tree, a kelp, a phytoplankton that is fixing carbon and is forming the base of food webs. So in the types of habitats sea otters live in, what we tend to see is that phytoplankton have much lower or more negative carbon isotope values, whereas kelps and things like seagrass have much higher or less negative carbon isotope values. So that's the carbon axis. And then nitrogen isotope values are often used to provide a metric for trophic level, because it turns out as you move up a local food chain, nitrogen 15 tends to bioaccumulate. So you end up with higher nitrogen isotope values as you move up trophic links in a local system. So then animals that are plotting in different quadrats of this space are generally, we can think of them as corresponding to feeding in distinct habitat and trophic combinations. Okay, so what we're going to do today is we're going to walk through some isotopic data sets from pre-fur trade sea otters, so archaeological specimens. We're going to look at 3 geographic regions: southeastern Alaska, northern Oregon, and California. And we're looking at these regions because they represent a really wide range of sea otter recovery trajectories and modern socio-ecological dynamics. So what do I mean by that? Well, southeastern Alaska, which is the sites we'll be looking at are from the ancestral homes of the Tlingit and Haida people, is an area where translocations of sea otters were very successful. And as a result, we have populations there that are doing really well and have almost completely filled in their historical geographic distribution. But what has also happened in southeastern Alaska is that as sea otter populations have re-expanded, there's been a lot of concern from local communities and local commercial fisheries about the impacts that sea otters are going to have on their livelihoods. And this has caused conflict with sea otter recovery and management efforts. Conversely, in northern Oregon, which is the ancestral homeland of the Tillamook and Clatsop people, this is an area where translocations did not work. The sea otters they brought there all either swam away or unfortunately died. So there are no sea otters in Oregon today. But what has happened is there's actually been kind of a grassroots movement, mostly driven in this region by tribal communities, to bring sea otters back. So really interesting how different this is from southeastern Alaska. And then lastly, in central and southern California, the sites I'll be showing data from are from the ancestral homes of the Chumash people. This is an area where translocation recovery was mixed, and it varied depending on northern, central, or southern California. So we have sort of a mosaic where some areas sea otter populations are doing really well, and other areas where they're still absent, such as there are still no sea otters north of San Francisco. Okay, so we're going to kind of go north to south here and start by talking about southeastern Alaska. And I want to draw your attention again to this last bullet point, which is this conflict between sea otter recovery, so sea otter sort of managers, and local communities and commercial fisheries. And there are three particular groups of macroinvertebrates that are sort of the flashpoints for all this. So when it comes to local communities, a lot of communities in southeastern Alaska rely really heavily on traditional and subsistence harvests of clams. So huge concern there with are sea otters going to decimate our clam populations. And then commercial fisheries in the region are mostly dependent on two species, Dungeness crabs and sea cucumbers. So we have a couple of different, species we want to look at when we're looking at isotopic data from zooarchaeological specimens in this region to maybe help us forecast which of these prey species might be impacted by sea otters in the future. All right, so here's a zoomed in map of southeastern Alaska. If you squint, you can kind of see the little dot there that says Juneau. The stars here now are denoting archaeological sites where we have sampled sea otters from. The biggest star is a site near Angoon, Alaska, which is where most of our specimens come from. This is a very important site because sea otters haven't quite gotten to Angoon yet today. So really exciting to see what the data there look like. So we have 6 sites in total. We think this represents about 38 individuals. We are pretty confident that these reflect locally harvested animals and not animals that were traded in. Happy to talk more about that after the seminar. And radiocarbon dating tells us that these sites were all occupied about 1,000 to 2,000 years ago. All right. And then because there are modern sea otter populations in Seek, we had the opportunity to compare our archaeological data to what's happening there today. I can't take bone from a living sea otter without doing some serious damage. So in this case, our samples come from whiskers, but that's fine. And we're focused on 2 populations in the north and south end of QU Island, which is also close to one of our archaeological sites. And then lastly, so that we can kind of address this question of how are sea otters going to impact clams, Dungeness crabs, and sea cucumbers, we also have a large data set of potential prey species in the region to help us contextualize our data. All right, so we're going to dive right into it. Here's our biplot again. I've left our lovely little clip arts to sort of help everybody stay oriented. Let's start with the prey data. So a lot is going on here, and what I want you to notice is where these key species are. So in the top right here, we've got our Dungeness crabs. The next highest one is the sea cucumber, and then those 2 red circles sort of moving towards the bottom left of the graph, those are clam species that are of concern for local communities. So where do our archaeological sea otters plot? Boom. Don't worry too much about the box at the top right. That is mostly for John. So John, rest assured, I have corrected all my data. Happy to talk about this more afterwards as well. But so what do these data suggest? Well, what we see is that sea otters in the past in this region really look like they were foraging mostly on clams, right? We do not see much evidence for them foraging on either sea cucumbers or Dungeness crabs. So how does this compare to modern sea otters? Well, here are the data from QU Island South. And you can see they're not quite the same. And actually, the little blue dots here, which are this modern population, are mostly clustering on top of red sea urchins. If we add in QU Island North, now we see again a really small clustering, this time over green sea urchins. So what we're seeing with both of these modern populations then is that they like sea urchins, right? This is kind of the classic sea otter ecological phenomena. And it is a pattern that we expect with sea otters recolonizing an area where they've been absent for a long time. Because remember, that without predators like sea otters, sea urchin populations can really explode. So what all of this is telling us is that the coastal marine food webs here in southeastern Alaska are in a really dynamic state. Things are changing a lot, and it's very likely that sea otters in the region are going to switch to other dietary items once sea urchin populations have declined. And if they do what they used to do, that means they're probably going to start focusing really heavily on clams. Okay, so I kind of jumped ahead of myself a little bit here. What do these data suggest about the future impacts of sea otters for fisheries and local communities in southeastern Alaska? Well, we kind of already talked about this, right? But here I'm showing just the archaeological data and then the modern prey data, and I've circled sea cucumbers and Dungeness crabs. And what is very obvious from this graph is, with maybe one exception, we don't really have any evidence that sea otters were eating these species in the past. Now, I do need to throw out a few caveats to this. So the first is that we're looking at bone, right? We're looking at the organic material in bone. And this records what the animal is putting in its mouth over many, many years, right? If you break your bone, it takes a long time to heal. It takes a long time to change the dietary signal that's recorded in bone collagen. So if sea otters in the past were only occasionally eating crabs or sea cucumbers, our tissue that we sampled is probably not sensitive enough to pick up that signal. The other possibility that's been raised is that Dungeness crabs and sea cucumbers maybe were not locally available in the past, either due to harvesting by people or due to over-harvesting by the sea otters themselves. We don't have an easy way to rule this out because, unfortunately, neither crabs or sea cucumbers preserve very well in the archaeological record. But today, what we do see is that both of these species frequently take refuge in really deep water habitats, which are outside of the range that sea otters can dive. So it seems unlikely to us that these species wouldn't have had that same behavior in the past, in which case they probably still would have been around, just in harder to reach areas. So, all of this to say that once sea otters have fully reestablished into seek, if they keep doing the same thing they used to do, we don't expect either of these species to be the bulk of their diet. And then, of course, very different story here when it comes to the potential impact of sea otter populations on local communities who rely on traditional and subsistence harvests of bivalves. And there's actually a really fantastic dissertation by Sonia Ibarra out of the University of Alaska Fairbanks, who did a survey and found that some communities in southeastern Alaska, over 50% of households rely on traditional harvesting of these species. And they've already seen declines in clam abundance following sea otter recolonization. So, we need to make room for both people and wildlife. And to me, this result really underscores the importance of community-driven approaches to managing sea otters and how important it is that these indigenous knowledge and ecosystem government systems are, considered and incorporated into our modern management decisions. Okay, that was seek. That was a lot, I know. So now I just like to start sort of working our way down the coast. So next, we're going to be looking at Northern Oregon and Central and Southern California, and then sort of comparing and contrasting what we see across all three of these regions. So we're going to start with Oregon, and Oregon is a really interesting area because, as I mentioned, there are no sea otters there today, but people are now interested in trying to bring sea otters back to Oregon. And the people that are really interested in doing, right, when you're thinking about where to translocate animals, the big question is, where do you put them, right? What kind of habitats? Advocates for bringing sea otters back to Oregon have talked a lot about how estuaries and soft sediment habitats would be good places for sea otters. And we do, in fact, find sea otters in these habitats in other parts of their range. And I'm showing on this map here is just a list of estuaries in Oregon. You'll see that there are quite a lot of them. So we sought to use the archaeological record to help us understand if sea otters were using estuaries in the past. Okay, so this is a zoomed in map here. You can see the Olympic Peninsula and the Columbia River running through it. And we have just two sites in Northern Oregon, Palmrose and Partee, and they're just south of the Columbia River mouth. So if there was ever a site where we were going to pick up an estuarine signal, it would be these two sites. Smaller number of samples here, but still respectable. As with Southeast, we're pretty confident these reflect locally harvested animals, and it's about the same time period as our sites in Southeast. And then because we have no modern sea otter data for comparison in Northern Oregon, what we did was just start by comparing pre-fur trade Oregon to Southeastern Alaska and California. Okay, so back to our familiar biplot at this point. Here are those same Southeastern Alaska data that we've seen before. And here are the data from Northern Oregon. So you'll notice there's some really interesting differences here from the Alaskan samples. So our carbon isotope values for Oregon are on average a little bit higher than Southeastern Alaska, which hints at some potential habitat differences. But of course, the most obvious difference is this variation in nitrogen isotope values between these two groups. So one possible explanation is northern sea otters in Oregon were eating at a higher trophic level. And I've teed you all up for this, right? With our nice, here's carbon, here's nitrogen, here's what these axes mean. However, when I was talking about nitrogen isotopes, I mentioned multiple times that this occurs within a local food web, right? And we actually think this difference is more a function of baseline differences among geographic regions, specifically in their oceanography. So southeastern Alaska lies within the Alaskan current, whereas northern Oregon is sort of at the tail end of the California current. And the California current is a strong upwelling system that brings up lots of isotopically heavy nitrogen to the surface that is then used by organisms. And the oceanography hypothesis becomes a lot more likely when we add in the data from California. And what you'll see now is that Northern Oregon and Central and Southern California have really similar nitrogen isotope values, at least on average, right? And this makes sense because, again, they're both part of the same oceanic current system, whereas Alaska has different oceanography. So that's what we think is going on with the nitrogen. I also want to take a quick step back and just give a little bit more context for the massive data from California I just threw up there. These are from my dissertation research that was published in 2020. There's 11 different sites here that represent 350 kilometers and about 7,000 years in California. I really wish I had time to talk about all this today, but for the sake of time, we just have to look at it here. So if you're interested, please let me know. I'd be happy to send you a copy of our paper. Okay, but yeah, so back to this regional comparison. And again, what's really interesting here is this range in carbon isotope values on this plot. So does this reflect real habitat differences by these populations in the past? Well, with these data alone, it's kind of hard to say. We've sort of exhausted the inferences we can make using just this coarse isotopic measurements. But we can get at these potential habitat differences in detail using what I like to think of as kind of the next generation of stable isotope techniques, and that is molecular isotopic measurements. So instead of getting a single value for a tissue, we're now able to analyze the molecular composition of the molecules, the isotopic composition of the molecules making up that tissue. So for bone collagen, which is what we're looking at in the archaeological record, right? This is a protein made up of lots of amino acids that are linked together chemically. And we can break the bonds between those amino acids, and then we can separate them using a gas chromatograph and some other fancy stuff. And that allows us to sort of segregate and then measure the isotopic composition of individual molecules. Right, so why do we want to do this? Sounds like a lot of work. It is. We do this because certain classes of these molecules, particularly essential amino acids, have really powerful inferences that they can tell us. So as the name implies, right, essential amino acids are molecules that we can't synthesize ourselves. We don't have the enzymatic machinery to make them. We have to get these compounds from our diet, or in some cases from gut microbes. We don't expect that in a sea otter. That's more if you're looking at like a cow or a ruminant. So What this means, a lot on here, but really I just want you to look at that last bullet point here. The carbon isotope values of essential amino acids are basically imprinted at the base of the food web, and then they're not really changed. So the isotopic value we're measuring in phenylalanine, which I'm showing here in this little box on top of the kelp, that carbon isotope value is going to be the same in the sea urchin, and it's going to be the same in the sea otter that ate that sea urchin that ate that kelp. So it allows us to kind of cut through the trophic noise and just look at what's happening at the base of the food web. And then in order to interpret these measurements that we're making in sea otters, we need a data set of marine primary producers from different habitats that sea otters might be spending time in. And so as another chapter of my dissertation, I collected and analyzed coastal algae from sites in Alaska, California, and actually northern Chile. And I focused on green, red algae, kelps, and then phytoplankton. So I'm going to be presenting data from these 5 essential amino acids that you see here. And because we now have 5 axes, we can't just look at a biplot. So we use a dimension reduction statistical technique so that we can sort of, every data point you'll see on this graph is kind of a composite of isotopic measurements of these 5 compounds. If you've ever done principal components analysis, it's a very similar idea. If you have no idea what multivariate statistics are, don't worry. Again, just remember that every data point on there includes measurements of 5 different amino acids. Okay, so here we go. Here's our data set of primary producers. I've added in these familiar clip arts to orient us to the different parts of this graph and what are relevant when it comes to sea otters. So kelps in the sort of bottom left here, the ochrophyta, that black circle. So they're sitting off by themselves, which is great. And then the blue here are now phytoplankton. And while it's probably obvious why kelp are included in this plot, I did want to note that phytoplankton serve as the base of the food web in a lot of sandy bottom habitats because things like clams are filter feeders. So what they're eating essentially is phytoplankton. So we're looking at phytoplankton as sort of our soft sediment habitat end member. Okay, so now we can plot our sea otters directly onto this space and use this to infer what habitats they were living in. So here are data from southeastern Alaska. And what you'll see is they're kind of in the middle. So there's a large proportion of individuals that are plotting with phytoplankton, but there's also a fairly large number that are plotting with kelp, which is really cool and something we wouldn't really have been able to infer just from looking at those initial bulk tissue isotope plots. Northern Oregon looks really different. And completely contrary to our expectation that maybe Oregon sea otters in this area were using estuaries, almost every individual that we sampled is plotting right on top of kelps, which is really cool. And then California, right, that huge range we saw in the carbon isotope values of archaeological California sea otters is reflected here. So these guys were clearly living in a huge range of habitats, from the kelpiest of the kelp forest to some very clearly soft sediment signals that we're seeing. Okay, and then the last thing that we did is we took these data and conducted a bootstrap resampling approach, and that's just randomizing the data that's in the primary producer sort of baseline model to see how sensitive where our sea otter specimens are plotting is to the specific samples included in this data set. So I'm going to plot the results here, again, going from north to south. And what you're seeing here in the blue are sea otter specimens that, regardless of the iteration of the model, we're always plotting right on top of phytoplankton. And the same thing in gray for individuals that were always plotting right on top of kelp. And then these checkered boxes represent what we're calling a mixed classification. So these are specimens that, depending on the randomized iteration of the primary producer data set, they were sometimes plotting with phytoplankton, sometimes plotting with kelp. And what I think this means is actually a real ecological signal that they were probably foraging in both kinds of habitats. So that's why we're calling them a mixed classification. And then, excuse me, taking a step back to What did we find across regions? Well, one thing that's really obvious is that kelps are important to sea otters no matter where you are, right? And so this really underscores that this classic picture we think of as sea otters living in kelp forests was as true back then as it is today. But what's really interesting here is Northern Oregon, where again, we really do not see much evidence for use of these soft sediment estuary habitats by sea otters in the past. So these data suggest that future translocation efforts maybe want to target kelp forests, or at the very least, take this into consideration. The caveat here, of course, is that unlike southeastern Alaska and California, our results in northern Oregon are really just from two sites that are very close together. So we're hoping in the future to expand this through analysis of specimens from southern Oregon and other parts of that coastline. Okay, so again, I know that was a lot to bring it home. I just want to remind you all what our sort of motivation for doing this work was, right? How can we understand what sea otters were doing prior to the fur trade, and how does this help us understand whether they've really recovered ecologically today? And what we found is that sea otters in the past lived in a really wide range of habitats, and they had broad dietary diversity. But there were really important biogeographic differences. In other words, it mattered whether that sea otter lived in southeastern Alaska or Oregon or California. And then more broadly, what we're hoping is that these data will help ultimately characterize whether modern populations, as they continue to re-expand and hopefully continue to recover, are occupying this historical niche space. And this will be critical information for conservation and management people to make decisions. And finally, to bring it all home, I just want to again, acknowledge that all of these insights are only made possible because of both the archaeological record and these powerful chemical techniques. And so to me, this really highlights the power and the importance of interdisciplinary studies as we try to find ways to balance the needs of people and wildlife on our continually changing planet. So thank you all so much for listening and for having me.
Emma, that was absolutely fantastic.
Thank you,
Thank
John.
you. Thank
You're a
you
bit
so much.
biased, but I'll take
I,
it.
I'm a bit biased, but I think I can see through this. It was really, really great. Thank you so much. So I wanna, we've got some questions rolling in, but I want to start with something that you said at the very beginning of the talk that kind of blew my mind, just the way you phrased it. Was like sea otters as like hummingbirds, right?
Mhm.
And so I've never thought about that before. And I'm pretty sure I know the reason why you said that, but they are these kind of hummingbirds in that they're not targeting carbs, sugar, right? But they're targeting what macronutrients to that help them stay warm.
Yep.
Is it?
So, yeah, that's a really interesting point, right? Because sea otters are totally weird in that they eat mostly fats and protein. They
Uh-huh.
eat very, very few carbohydrates.
Right.
And part of that is probably to help keep them warm, right? Because of course,
Right.
fat is a fantastic fuel. It's easy to burn it. So they definitely target fat-rich prey items when they can get their hands on them. And that's part of the reason they love sea urchins so much, is that sea urchins are basically gonads and a bunch of fat.
Hmm.
And that's what a sea urchin is. And so they're just these little energy balls for sea otters.
Very interesting. And protein, right? When you're digesting that, that keeps you the warmest. Is that true?
Actually, I'm not sure about that. So I don't want to say yes and be incorrect.
Okay.
But both of those two are better. They're more energy dense than
Yeah,
carbohydrates, for
for
sure.
sure. Ah, it's so interesting. It's fascinating just the way you said that. Um, One of the other questions that I have, um, so with Oregon, there have been previous translocations, right, of sea otters that have failed.
Yes.
And can you talk a little bit about that history and then how you see this playing into, where they come from, where these otters come from matters in terms of success and all of that?
Yeah, absolutely. Thanks for giving me the opportunity to bring that up because it's a very complicated but really interesting story. So, firstly, I should acknowledge that any translocation is just inherently difficult. Like
Yeah.
whether you're translocating a wolf, a puma, a sea otter, animals don't like to be moved around against their will, right? So we typically expect low success rates when we're doing translocations. But the ones we saw for sea otters in Oregon were just particularly bad. And you mentioned sort of where they came from. So the translocations in Oregon, um, I'm not going to say the year because I don't remember it exactly and I don't want to be wrong.
Mhm.
But they came from basically the far western Aleutian Islands. They were translocating sea otters from way the heck in the North Pacific to
Right.
Oregon. Um, and there's been a lot of interesting discussion actually over the Oregon population in particular, because a lot of people thought we actually, sorry, take a step back here. Sea otters are a single species, but there's three different subspecies of sea otters. There's the northern sea otter, the sort of central Alaskan sea otter, and then the California sea otter, different subspecies. And a lot of people have wondered if there was a fourth subspecies in sort of the Oregon, Washington area, or if not, were those animals more closely related to California or were they more closely related to Alaska? And maybe that's why this translocation didn't succeed. So there's been a little bit of work done on this by Hannah Wellman, one of my colleagues. She actually found that the now extirpated population in Oregon was most closely mitochondrially related to southeastern Alaska. So,
Mhm.
um, but the problem now, of course, is that the southeastern population came itself from translocations. So we've lost a lot of genetic diversity in sea otters that we're sort of struggling to get back. Um, but yeah, and this also then, this all falls under kind of another question, which is when you're translocating sea otters, should you translocate the most genetically similar individuals or the most
Right.
ecologically similar individuals? I fall on the ecology side of that debate. So to
Hmm.
me, I'd say you should take sea otters that maybe are from kelp forests and try to translocate them into areas of Oregon that still have kelp today. But, I'm not a on the boots on the ground manager, so I'll leave those decisions to the people that know that.
I think it's a really great point, Emma, and it's actually a really great segue into one of the questions that we have here, which is, are there kelp forests in Oregon, or have the sea urchins had a huge effect there? What's that like?
The fantastic question. The answer, of course, is nuanced. There are some, but there's not a lot. They're very patchy. So there's like a little patch of kelp and then a lot of nothing and then a little patch of kelp. The problem, which you just alluded to, is that we haven't had sea otters in Oregon for, 150, 200 years.
Mm.
Um, and it used to be that predatory sea stars kept sea urchin populations in check. But I don't know if anyone here is familiar with the wasting disease that decimated sea star populations in the Pacific about 10 years ago. Um, and what happened is once the sea stars had been removed from the system, sea urchins exploded and have overgrazed a lot of kelp. Um, so yeah, but sea otters might be able to help fix that problem, right?
Mm.
Yeah.
Pretty interesting.
Mhm.
Um, we've got a broad question here. So, how do paleo-ecological and archaeological methods overlap, or is there differences? This person says, I'm an archaeology student interested in ecology, and I'm curious about the field of paleoecology.
Oh, that's a great question. I think it really mostly depends on who's doing the paleoecology and
Sure.
who's doing the archaeology.
Yeah.
Um, I have colleagues who are archaeologists by training, but really think about things in a very ecologically minded way. Um, there is a lot of overlap between paleoecology and archaeology. I think one of the big differences is that a lot of paleo people come from a geology background. And so when you're looking at like excavations in paleoecology and stuff like that, they excavate in a completely different way, right?
Right.
Yeah, it's like it's not these little 10 centimeter increments. It's like, here's this strata, here's this strata, here's this strata.
Right.
So that can be really complicated when you're trying to compare and contrast archaeological and paleoecological data because they're generated in very different ways. Uh,
Yeah, and the conceptions of time, right, too, just of like the amounts of time that people are dealing with. In my experience, people get very frustrated with archaeologists sometimes because we're so nitpicky about time. So, like for instance, here in the Southwest, if we want to date something and we have
mhm.
dendros, we could get to the year that it like a structure was built.
Right.
But that level of like thinking about what is happening can sometimes be frustrating for others, and vice
Absolutely.
versa, right?
Yeah. And the other big difference is, you know, people, I don't like this phrase. People say, throughout like, oh, you're looking at everything through a human filter. You know what I
Uh,
mean? It's the human filter of archaeology. You're only looking at what people were choosing to go out and get and bring home. And that's true, but people are generalists, like the most generalist species, right? So, there's a lot of reason to think that like they're doing a good job of sampling for
mm,
us. And the other part of this is paleoecology comes with its own set of filters, right?
mhm.
Um, that are both taxonomic and environmental. So I'll leave it at that.
Cool. Okay, we've got a kind of a specific question here, and I'm going to read the whole thing. It's a little long, but it's good. So this person says, "Fascinating presentation. Thank you for the detail. I'm sitting in Thorn Bay, Alaska right now on Prince of Wales Island. I have several questions, but I'll keep it broad. I know that here on Prince of Wales, we have a problem with paralytic shellfish poisoning, or PSP, that heavily
Mhm.
curtails subsistence harvesting of clams.
Yeah.
So, does PSP affect sea otter diets? Are attitudes towards sea otters more positive in Southeast Alaska communities with PSP issues? This is beyond the scope of your work, but do you think there's hope for sea otter predation helping to combat European green crab invasion? Thank you again. I definitely plan to read more detail about the sites you sampled. And yeah, so
Awesome. Yeah. Well,
yeah.
hello from Prince of Wales. What an amazing site in southeastern Alaska. Yeah, and there's been some people that have done some really fantastic work on modern sea otter populations in the region using stable isotope analysis. Um, okay, so firstly, the paralytic shellfish poisoning. Yeah, so that's a huge issue across the range, actually, not just in southeastern Alaska. Does it impact sea otter diets? The short answer is we're still trying to figure that out. It doesn't look like sea otters are smart enough to avoid shellfishes with PCP, and it actually can cause health issues in the sea otters themselves. So in Central California, actually mortality rates have increased quite a bit with sea otters due to that toxin as well as sarcocystis, which is famous from cat feces and stuff like that. And what we're still trying to figure out is how PCP gets to the shellfish, like through which route. We know it comes from land and gets into the water column, but are there specific habitats that make both clams or sea otters more susceptible to being exposed to this pathogen? So there's a lot of work still to be done on that. In terms of the sort of how people feel about sea otters in southeastern Alaska, it is really mixed. So there are a lot of people that do see the benefit, particularly for controlling invasive marine species, like you mentioned, the European green crab. But right, there are understandably also a lot of people that are really concerned about the impacts that sea otters are going to have on their own lifeways and livelihoods. So it's a really thorny issue in southeastern Alaska, and I would say that overall, the reception towards sea otter recovery is a little bit more negative in Southeast than it is in like Oregon or California, for sure. So, but yeah, we're working on it. And I think a big thing that will help is providing more agency, particularly to Indigenous communities in the region, to manage sea otter populations in their traditional ways, to both protect their, clam beds, their clam gardens, their harvesting areas, um, and that will sort of, hopefully help with that. So I'll leave it at that.
This person also, Natalie Madison is their name. They just posted it in the chat. Said, I think you'd be a wonderful presenter at the annual Sitka Whale Fest in Sitka, Alaska. Emma, do I have your permission to drop your email address in the chat?
Absolutely.
All right.
Sure.
All right. One last question. I think this will be a pretty easy one, Emma. How far up rivers did otters roam? They can survive in both types of water, right? Sea otters, what's that like?
Ooh, that's a great question. I don't think I know,
Hmm.
actually. Yeah, so I guess the answer is I don't know. They can survive in both types. But I think you see the inverse more than you see this. You see river otters in marine systems more frequently than you see sea otters in rivers. Part of that is they're really not good at catching fish. They really suck. Yeah, they're too fast.
Too
Um,
fast.
and that's mostly what there would be to eat in freshwater. But yeah, in terms of how often they do it, I don't know. It's a good question.
All right. All right. I want to be respectful of your time. Emma, thank you again so much for such a fantastic presentation. We've got all kinds of thank yous rolling in and just people talking about what a great job you did. So thank you very much.
Thank you, everyone. Yeah, really happy to be here, and please don't be shy about reaching out if you have follow-up questions.
All right. All right. Have a good one, everyone. Bye.